Institute for Mathematics and its Applications here at the University of Minnesota. And the IMA Institute is your host in these Math Matters Public Lectures. Today, I'm very pleased to open up our season for the year. It'll be a season of 4 lectures as usual. And our first one is given to us by Professor Bernd Sturmfels. Bernd is somebody we know very well. In his early career, he was educated in Germany, did most of his graduate study in Germany, but then went to Seattle. And he finished in 1987 with actually 2 doctorates, one from the Technical University in Darmstadt in Germany and one from the University of Washington in Seattle, with theses relating to combinatorial geometry. Then Bernd made a big step, which I think was a big step in his career and also important for us, because Bernd had an advisor who said he should check out an exciting new math institute. This was back in 1987, that was getting founded in Minnesota about 5 years earlier, called the Institute for Math and its Applications. And Bernd came and joined our postdoctoral program. We had a program that year on applied combinatorics, and that had a lot of influence, I think, in setting Bernd's future direction. He became a faculty member in Cornell 2 years later, and then in 1995 he moved to Berkeley, where he presently holds a joint position as professor of mathematics and computer science. He's a highly honored researcher, a very influential mathematician. He has many awards in his career, starting with a National Young Investigator, a Sloan Fellowship, a Packard Fellowship. He's been the von Neumann Professor at the Technical University of Munich, a senior scholar, Clay Senior Scholar at the Park City Institute, and many other things. He's a prolific author and a very successful one. He's written he's written 8 books. He's edited about half a dozen more. He's the author of 150 papers. Besides their mathematical content, his writing is superb. He won the Lester R. Ford Award for Exposition in 1999. And just recently, during our IMA program, when I was visiting a lot, he and his collaborators had a paper in the BMC Evolutionary Biology Journal. And although this paper probably had 2 orders of magnitude more mathematics than the typical paper published there, it was the most downloaded paper in the Evolutionary Biology Journal in the month of May 2007. Bernd is also a wonderful organizer and a wonderful mentor of young people. He's had more than 20 PhD students. His PhD students already have PhD students. And I'm going to take an example to read an email that I was CC'd on a year ago when Bernd was an organizer for us. So Bernd came back after having been one of our star postdocs and came back and was a lead organizer of our program last year in applications of algebraic geometry. And he sent this email to 2 of our new young postdocs who had arrived for that year. So if you're a professor of math and you wonder what does it take to be a great mentor, at least I felt I could learn a lot from this letter. So he wrote, "Dear Hannah and John, congratulations, you've won. The 2 of you have been selected as organizers for the event Sunday with Mike and Bernd, to be held at the IMA on Sunday, September 24th. Mike Stillman and I, Mike was another organizer, Mike Stillman and I will be in town, and we'd like to have an informal working day with all the young people only at the IMA. By this we mean all the IMA postdocs and graduate students in residence and interested young people from the University of Minnesota. All the rest of the details are up to you. Our only specific suggestion is we start early enough, like 10:30, and we end with dinner, including a beer, together. As I said, details are up to you. I think both of you knew what kind of style is intended. They know because Bernd had done this before with them at other places and another time. People can tell the others what they're working on, discuss, ask questions, ask about anything they heard at the IMA workshop they didn't understand. And of course, Mike and I will be quite prepared to suggest problems to work on and make you guys work hard. The main point is to keep it informal and to allow only young people to participate, except for Mike and me, both of whom are truly ancient. So please go ahead, reserve the room, spread the word. I'll see you soon, Bernd. Okay, besides being a wonderful scientist, a wonderful author, organizer, and mentor, he's also a great lecturer. I could read you lists of distinguished lectures he's given, but I won't. I'll simply let you judge for yourself as he gives us a talk on algebra, statistics, computation, and biology. Bernd Sturmfels.
Scott, for the very nice introduction. It's always nice to be back in Minneapolis. What I remember from my postdoc days is that there are only 2 seasons, right? There's summer and winter, and I was very lucky. I think I just hit the one day changes. So I'd like to tell you about, this was the title I originally gave, Algebraic Statistics for Computational Biology. And the talk will be about this picture. So you've seen the picture on the flyer and on the way in. And I hope when you leave tonight you have a better idea of what the picture is about, what's shown on the picture. So, well, let's start with some definitions. Math talks start with definitions. So we are blessed with online dictionaries. So you could go to Google and pick one of the online dictionaries and you can ask, what is biology? Now biology, according to one of those websites that I picked, oops. Are we okay? So biology is the study of living organisms. And there's a little person up there. So biology concerns living organisms like him. Statistics, according to the same website, is the science concerned with the collection, organization, analysis, and interpretation of data. And to make sense out of these data, we'll see very high quality data in this talk. And the role of statistics in the data that I'm going to show you is not so much to sort of extract signal out of the data, but to just make sense, to understand the data. Finally, what's algebra? So the same dictionary says that algebra is the part of mathematics that deals with generalized arithmetic, and we will also see generalized arithmetic in this lecture. Okay, algebraic statistics was in my talk, in my title. So then what is algebraic statistics? And the online dictionary is remarkably silent. There is no dictionary definition yet. So this term exists, and it was coined by a group of statisticians, Europeans from Italy and England. And they were interested in applying a particular method from algebra. Rotner basis is a method from algebra to a specific field in statistics called design of experiments. So they got very excited, they pursued this, and they ended up writing a book called Algebraic Statistics, Computation, Algebra, and Statistics that appeared in 2000. And I borrowed the title from them. So Algebraic Statistics, the term is taken from them. Now, that was the first book on algebraic statistics, and here's the second one. So sometimes when people go around and give these lectures, really what they're trying to do is to hawk their book. So there's a second book called Algebraic Statistics for Computational Biology, and this was put together by a collaborator of mine, develoP.M.ental, and myself. And this grew out of a graduate seminar that we did in Berkeley in the fall of 2003. And we had lots of wonderful young people, grad students, postdocs from math, other fields, computer science, statistics, integrated biology, and so on. And the seminar went so well that we decided at the end to put together a book. So Leo and I were the old guys, so we wrote part 1. And part 1 has 4 chapters. And they have these ridiculous titles, right? They're called Statistics, Computation, Algebra, and Biology. And it's of course ridiculous because you cannot possibly try to do justice to any of them in, say, a 40-page chapter. But basically what's happening there is we just sort of give a snapshot, an introduction of the theme from these fields. And then the rest of the book was written by the students and the postdocs. So each group of 2 or 3 young people picked the topic, and they wrote these chapters, and some of these chapters, they look like biology, vertebrates and flies, looks like biology, and some of them are more mathematical or more statistical. But in any case, on the cover of this book is this picture, and I don't want to bore you with the details, what's in the book, I just want to tell you what's the picture. Who is the girl? Now, there she is. It's actually very interesting. So we had originally a drawing, which my daughter helped draw, and that was sort of the California version of the girl with blue jeans, an American girl, and this went then to Cambridge University Press, and that's what came back from England. And at the beginning we were a little shocked, but in the end we kind of liked it. like the pink skirt. So who is she? Well, she's Diana. And Diana makes Diana sequences. And, well, how does she do, what kind of sequences are they? Well, for short, we also abbreviate them as DNA sequences. And these are sequences over a 4-letter alphabet, A, C, G, T. And what she does in her pink dress, she has a tetrahedron, a tetrahedral die, that has 4 sides. Right? So it's a tetrahedron with 4 sides. And on the 4 sides, you have the letters A, C, G, and T. Now she rolls this die. Now, of course, when you have a tetrahedron, it's going to land face down. Right? So you're not going to see a face up. So you look, what face lands down, and then that's an A, a C, a G, or a T. And she does this many times, and then maybe that's the record of what happened. Now, these symbols in biology represent the basic building blocks of carbon-based life. So they are the molecules adenine, thymine, guanine, and cytosine. And that's what she does. She basically rolls the die and makes sequences of these letters, and they represent these molecules. We talk about the human genome. So the human genome, to first approximation, is a very, very long word over the 4-letter alphabet A, C, G, T. It has about 2.8 billion letters. These letters are also called DNA or Diana bases. And this was actually a big accomplishment. In 2001, the first sequence was published. The human genome was sequenced in 2001. But then, if you write a book or a term paper, well, it needs many revisions, right? You get criticism back from your professor, you have to revise it and rewrite it, and that same thing happened here. So it was rewritten about 35 times. And then in 2004, there was another paper that says, finally, we got it, and it was considered to be finished. Now, of course, why is there one human genome? Well, of course, each of us has a different genome. We have our own genome. But the differences between any two of us is so small, maybe every 1 in 10,000 or 20,000 letters, that's negligible relative to the error rate is even less than that. But we will neglect that and sort of think about having the same genome, and that's what people do. Now, one thing that the genome does, it contains genes. So what's a gene? The gene, for the purpose of our discussion, is a subsequence of the genome. And those subsequences, they code for protein. That's what they do. And interestingly, we still don't know, the exact number of genes. And perhaps it doesn't even matter. Suffice to say, it's between 20,000 and 25,000 genes in the genome. Now, the genes, they make up less than 5% of the genome, so 95% and more are between the genes. And until quite recently, people thought about that as sort of dark matter or junk DNA. And it's in the last couple of years that a lot of study has been focused on those 95% on the stuff between the genes. Well, let's go one step at a time. Let me give you an example of a gene. So I'm going to show you an example of a gene, and I'm going to show you how you, tonight, when you log back in at home, how you can read the human genome. Okay. So the gene I'm going to show you is called, as the abbreviation, BRCA1, and it's a gene that's linked to breast cancer in females. So it's called the breast ovarian cancer susceptibility gene. And females who have a change in this subsequence are more likely to develop breast cancer. So it's a medically very important gene. Okay, so if you want to look at the human genome, and maybe you would like to find this or any other gene, you go to a point of entry, and a good point of entry is at the UCSC, University of California at Santa Cruz. So you go to genome.ucsc.edu, and that's what you get. You get a web browser, Genome Bioinformatics, and you click on the genome browser. So I'm going to take you through 5 steps now. So you click, and then you get this gateway, and then you can pick, the clade, which genome. So we are vertebrates, then you can click the human genome. But when we first wrote our book, there were about 9 genomes fully sequenced. I think in the meantime, there are 50 or 60 available. So you go there, and then actually 2004, it was considered finished, but not quite. So there's actually a newer version. The one that's up at Santa Cruz now is the March 2006 version. And then you tell the position or search term that you're interested in. Now, in this case, we're looking for the gene. We know the abbreviation BRCA1. You could also type in the raw coordinates, if you know them. Or maybe if you don't know, you can just type in a medical term. So maybe you can type in breast cancer or cystic fibrosis, and then see what comes up. You click on Submit. And then pops up a window, and it shows, genes, positions in the genome. And you have to click around a little bit. We're going to pick the third one. So you already see here there are these long numbers. And so here it says, it's linked to this medical term. And it says CHR17. Now the way this genome, this very long word is organized, it's organized by chromosomes. So this particular gene lies on chromosome 17. And we're going to zoom in now on position 38,468,876, ending 38,530,994. There you go. Now another window pops up. And this is kind of the hardest one to read for mathematicians. So biologists, people who study molecular biology, they're used to, they get trained to read these kind of maps. But this is basically a map of the genome, so the annotation. And you can see a couple of things. I'm not going to explain this, but these columns correspond to sort of positions. Then down here you see other organisms like mouse, dog, opossum, platypus, chicken, and so on. So these are other organisms. And then these little spikes, they tell you, the relationship between the human genome and the corresponding piece in these other genomes. And then there's other stuff, right? There's a gene bank, RefSec genes. So let's not worry. Let's move on. And let's actually click at the raw data at the DNA. And what I'm going to show you now is I'm going to zoom in on a region that corresponds to what's circled in red. And you can see 2 little tick marks. Do you see these little 2 black marks inside the red region? So I'd like you to remember that there are 2 tick marks in the red region. Okay, you click. There you go. There you go. So that is the actual sequence. So it took you about 4 or 5 steps. So try it tonight. If you can't sleep, you can maybe read the human genome. Okay, so UCSC, it's the genome browser at Santa Cruz. Just say Genome Santa Cruz. Okay, that will take it. Okay, now what about these data? Well, first of all, these data, as I said, are very accurate. So this is basically, this is our common genome. So all of us agree, except maybe in 1 or 2 spots here, but basically this is our sequence in this region, right? So on chromosome 17, starting in position 38,464,686, right? Going to 38,473,000 and so on. So that's the actual raw coordinates. Now, what I promised was that we're going to look at this gene, BRCA1. So where is the gene? Well, it turns out there is the gene. So this tiny piece, well, the first thing I should say, I said genes are subsequences of the genome. Well, that's true, but I didn't say that they're connected. In fact, they're not. Right? So genes, in fact, are disconnected. They have several pieces, and these 2 little boxed subsequences, they're called exons of this gene. And that's sort of where the information is as far as coding for protein goes. Now, where's the math? Where's the computation? So one task that mathematicians and computer scientists and statisticians got involved in this is to predict where are the genes. So So gene prediction is the problem of seeing this data and finding these two subsequences as the ones that are coding for protein. So this is an example of a problem, a sort of mathematical problem in computational biology, to go from this input to this output. Now, how does this work? How do you go and find these subsequences? And I'm going to not tell you exactly, except I'm going to give you an analogy, and it's an analogy of something that you are using every day. So here's the analogy. And the analogy that surprisingly similar is the did you mean feature in Google. Okay? Well, so let's say you go to Google and you type in some term, and in our alphabet, right? Well, what's our alphabet? Well, basically, maybe let's say we have 80 characters, right? We have lowercase, uppercase, space characters, special characters, blanks. So you type in some sequence of our alphabet, so TR space, OPICAAL space, GEETRY. So type this in. Now, from Google's point of view, I've given Google a sequence, and Google is supposed to make sense of this sequence. Well, of course, there's some differences between, this red sequence that Diana gave us, right? Diana gave us this red sequence to me, or to us, and the black sequence that I gave to Google. Well, one difference is Diana's sequence was red and my sequence is black. Another difference is that my sequence is short and Diana's sequence is long. And another difference is that my sequence is over a big alphabet and her sequence is over a small alphabet. But other than that, and we're giving a sequence to Google, we click, search, and then Google comes back and says, did you mean tropical geometry? Now that's amazing, right? That's exactly what I meant. That's incredible. I meant to say tropical geometry when I typed this stuff. So how did Google know what I meant? Right? That's incredible. Now how does this work? Well, what Google does, it annotates my sequence. Google calculates the most likely annotation or most likely coloring of my sequence. In this case, I'm going to illustrate this with 3 colors. And the colors are keep the letter or keep the symbol, change the symbol, or delete the symbol. So here's my sequence, my input sequence. And then among all possible colorings of my sequence, Google calculates the most likely color. Now, most likely here means that Google has a particular statistical model implemented, so something called a hidden Markov model. And in this model, a particular choice of parameters. So we have a fixed statistical model and a fixed choice of parameters. And with respect to that, Google calculates the most likely coloring with 3 colors. And so, keep the green stuff, delete this red one, and then here make a change, and then some subsequent calculation finds what to replace it by. But you can see the analogy, right? So in the sequence, the large sequence that Diana gave us, we're also, in gene finding, calculating the most likely coloring, right? There are 2 colors here, boxed or non-boxed. Interesting, uninteresting. Exon or non-exon, intron, whatever. So that's what this program does. There's a certain statistical model, something like a hidden Markov model, a certain choice of parameters, and we're calculating the most likely coloring of the red sequence with 2 colors. Now what about these parameters? Now, in Google's case, they know the parameters fairly well because they know the English language, the use of words. They have a lot of data because you and I type in every day and they collect data whether we're happy or not. So they have fairly accurate information about the parameters and what would make us happy. In the case of computational biology, it's generally more difficult because, well, there aren't that many genomes. And so it's harder to have accurate information about the parameters, and that's where some of the techniques come in that I'm going to describe. Okay, maybe this would be a good moment to pause. Quick question or comment, correction on the biology. All of you guys take molecular biology, you know this better than me. Okay, well, let's go back to Diana. Well, you know, Diana, she really doesn't have one die, right? If you remember, when you walked in on the flyer, she wasn't having just one tetrahedron, she has two tetrahedral die. And there they are. She has a red one, maybe, and a blue one. And maybe she generates not one sequence, but two sequences. So let's pretend Diana walks around, and she rolls the two die at the same time. So each time, maybe the first time, she'll see the red T and the blue T, and so on and so on. Sometimes they differ, right? Sometimes the red one makes an A, and the blue one makes a T. She does this maybe 60 times. And then what she produces are two sequences. And these sequences are aligned in the sense that, you know, every red character has a corresponding blue character, and vice versa. Okay, you can imagine, right? So there she is. She has a pink skirt. She rolls the die. She makes two letters. She does it again. She does it again. And she makes two aligned sequences. Now, what kind of questions could we ask about these sequences? Well, one question we could ask is whether these two sequences are independent. And let me explain what I mean by that. So let's suppose, just for simplicity, that the subsequent, you know, every dice roll and the next dice roll is independent from each other. What we can do is we can summarize the data. We're going to summarize these two sequences in a 4x4 table of natural numbers. And the meaning is that 9 times there was a red A on top of a blue A, 4 times you had a red A and a blue C, and so on, and 13 times we saw a TT. So let's regard this as our data. Now the question I like to ask is, when she has these two die, are they independent, or is there maybe a string between them? Is what happens with the left-hand die, is it, you know, correlated in some way to what happens to the right-hand die, or not? And we'd like to answer, try to assess this question by looking at this data summary. Now there's sort of two ways of phrasing the question. So statistics question is, are the two sequences independent? Right? So that's the question I would like to ask. Now it turns out that there's a corresponding algebra question. This is a linear algebra question. Namely, the question is whether this matrix of numbers is close to a rank 1 matrix. And I'm going to explain this in mathematical terms, what this means. So I claim that these two questions are essentially the same question. Okay, a little bit of math. So here we're talking about statistical models. And if you open a textbook in statistics, so the textbook, a textbook definition of statistical model, sometimes it's a family of probability distributions. Now family doesn't mean mom and dad and you. Family means a set, you know, a subset of some space. And these sets in the situations we describe here are subsets of some maybe n-dimensional space. And they come in parametrized form. So here we have discrete data. So statistical model for discrete data is a function that goes from a space of parameters, which I think of as a d-dimensional space, to probability distributions on m possible observations. So m is the number of observations we can make. Maybe sequences of a certain length. And m is a large finite number. So maybe m might be, you know, 4 raised to the power 2.8 billion. Right? The number of possible genomes. Now D is also a number, but that's a very small number. So these are parameters. That might be 3 or 5 or 7. So think of D as a small number and M as a big number. And we have this map that says the probability of observing a particular, so the ith coordinate says the probability of observing, making a particular observation is a function of these parameters. Now, of course, we want to get probabilities. The vectors we get in M space should be non-negative. They should sum to 1. And so there's some legal space of parameters. Now, in the book, we're assuming that the coordinates of F are polynomials. And in that case, the families, these models, are called algebraic varieties. Now, the picture that you might want to have in mind is the following. So there's some low-dimensional space of parameters. That's this little segment here. So there's some model parameters, and Google knows their parameters very, very well. But in biology, in computational biology, we don't really know the parameters very well. Now then we have this model, F, this map, that maps this very low-dimensional parameter space to a very, very, very high-dimensional simplex. So think about this triangle as a, very high-dimensional simplex. The image is this little red wiggle. So this red line here maps to this red wiggle. And the data, they are a point in here. So what is the data? Well, you go out there and you count how many times each observation was made. Right? So the first observation was made 9 times, the second observation, and so on. So you get a vector of length m, non-negative integers. You divide by the grand total, and that's this point. That's the data point. And the kind of inference we would like to make, maybe, is to ask, is this model a good explanation for the data? Is the data point close to this model? And one technique for doing that is called maximum likelihood estimation. And here is the explanation for this little independence model. So I said that independence in the statistical sense is the same thing as being close to rank 1. Indeed, so in that simple model, there were 16 possible observations, right? AA, AC, AG, AT, down to TT. So those are 16 observations. Now, when Diana hops around with her 2 die, these are not fair die, right? They are loaded die. So with one of these tetrahedra, the probability of seeing an A is not a quarter. It's some other number, but we don't know, right? The problem is we don't know what's the probability. She knows, we don't. So therefore, there's a parameter. So alpha A is the probability that Diana's red die rolls an A. We don't know that. Alpha C is the probability that the red die makes a C, and so on. And beta T is the probability, the unknown probability, that the blue die comes out to be T. Now it looks like there are 8 parameters, right? There are 8, the 4 alphas, 4 betas, but they're linked by 2 equations. Now if these things are supposed to be probabilities, well, they should be non-negative numbers, and each of them should sum to 1. The alphas should sum to 1, and the betas should sum to 1. So therefore, really, it looks like 8, but there are only 6 parameters. Okay, so then, the model that I was describing here, we have 6 free parameters, the alphas and the betas, and then we map this to these 4x4 tables of probabilities, and the map is linear algebra notation, so alpha, we'll take this alpha transpose, let me get this right, so this is a row vector multiplied with the column vector. If you multiply a row vector with a column vector, you get rank 1 matrix, and that's exactly the condition for independence. So independent means to be in the image, the probability is factor, so independent means the probability, if the weather tomorrow is independent of whether the bus is late, and it says the probability that it snows tomorrow and the bus is late is the product of the probability that it snows tomorrow times probability that the bus is late. Okay, so that was the independence model. Now, that model and what I described Diana doing is hopelessly, it's irrelevant. It's too simple, everything is too small. I mean, if we have only n equals 16 observations, that's not a biologically realistic situation. So let me take about 10 minutes and describe a slightly more realistic problem, a slightly more realistic scenario, and then we'll sort of slowly wrap up. So here are some real data. There's a red sequence and there's a blue sequence. And these are actually sequences. So this red sequence actually was on this big chart that we saw earlier. So this is in this region that we looked at earlier on the genome, in the human sequence. Now this blue region over here is a corresponding sequence from the rat genome. And let's pretend that Diana made these two sequences. And then let's see what kind of questions we could ask about these two sequences. Now the very first thing that you observe is that they don't have the same length. Right? So whatever Diana did to make these sequences, it wasn't what she did 3 minutes ago. She did not, take dice and roll them at the same time. She must have done something slightly different, right? Because she generated two sequences that don't have the same length. Now, they're not far off, right? The red sequence is 3 letters longer than the blue sequence, but they're not the same length. So these are the observed data. Now, from the observed data, using statistics, algebraic statistics, we like to calculate the hidden data. And here are the hidden data. So there's some program that takes this input, it does something, and it makes this output. Now what is this output? Okay, well the output, what happened was the red sequence got aligned with the blue sequence, meaning we put one red character on top, precisely on top of a blue character. But of course, they didn't have the same length, right? The red sequence was 3 longer, so to put, correspondence between the red, characters and the blue ones, you have to introduce 2 blue spaces. And that's what we did. Right? So there's 3 spaces that inserted. But, you could also maybe have 4 blue spaces and 1 red space. Or maybe, 11 blue spaces and 8 red spaces. And there are many ways of doing this, and these things are called alignments. And what we calculated here was unoptimal alignment. And let me tell you what this optimizes. So I put a star whenever the red letter agrees with the blue letter. Right? So you can see there's a star, that's good, right? Whenever the red one matches the blue one. And there are many stars, right? So that's good. So this is an alignment that has a lot of stars. And from that information, we did something else, namely, we capitalized some of these letters, right? So now in the input, that wasn't the case, but in the output, we actually sort of highlighted some of these letters that capitalized, and those are actually the exons from the breast cancer gene. Right? So these are actually the corresponding sequences in the human genome, and this is the corresponding exon in that same gene in the red sequence. And just by looking at this, you can see there's a higher degree of agreement, a higher degree of conservation in this genic region. And then, okay, you can see other things, if you look a little closer, but I'll leave it at that. So this is called an alignment. Okay, so I would like to take whatever is left for my promised 10 minutes, maybe 7, to tell you the statistical model, explain to you the statistical model that takes us from a pair of sequences like this and that calculates a good alignment. Well, that will take us to this part of the picture. And that will be the last part of the picture. and that will slowly lead to the conclusion of the talk. So I explained who she is, I explained what that is, but I didn't tell you yet what this part of the picture is. Okay, so I'd like to explain to you that the hidden data corresponds to a path from start to finish in this diagram. And this diagram is called the alignment graph. So imagine, there's a little diagram like this, and you start here, and you want to go from start to finish. And the steps you can take is you can make a step north, you can make a step east, or you can make a step northeast. Right? And you want to go from start to finish. Now, okay, you could go, east, east, north, east, north, north, east, east, or something. And that's one path from start to finish. Now, if you think about it for a moment, if you're given two sequences, then the possible alignments of those two sequences, the ways of putting red and blue spaces, so as to put them on top of each other, are precisely the path from start to finish. So let me explain this by way of this example. So suppose the two sequences that we've given have length 5 and length 4. So the first sequence that she makes is the red sequence G, T, 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 A. The second sequence, the blue sequence with the second die, is C, G, T, G. Now there are many ways of aligning them. Here's one possible alignment with these stars. And I claim that the many possible alignments correspond exactly to the many possible green paths from start to finish. Well, indeed, right? You're right. There are one sequence, vertically, the other sequence horizontally. And whenever you take a diagonal step, that means, you have a red and a blue letter together. Whenever you take a eastern step, you only have a red letter and a blue space. And if you make a northern step, then what she does is, in the northern step, she generates a blue letter and a red space. And that's what she does. In her pink skirt, she jumps on this alignment graph. So Diana jumps from start to finish. She jumps from start to finish. And this alignment graph is exactly this picture, this part of the picture. And that's what she does. Okay, so let me demonstrate. So she has two die. She has the pink skirt. And she is somewhere on this graph. She starts at start. Then she makes a decision. She makes a decision. Am I going to take a northern step, or northeastern step, or eastern step? Now maybe she says, I'm going to take a diagonal northeastern step. While she's taking the step, she rolls the two die, and she makes a red letter and a blue letter. Now if she decides to make a northern step, she only rolls the blue die, but not the red. If she takes an eastern step, the other way around. Right? So if she makes a diagonal step, she rolls both die. If she takes, a northern or eastern step, she only rolls one. So here again, so as Diana jumps on her graph, she throws the dice, and she randomly generates the observed data, which are two sequences of different length. So for instance here, in this example, so Diana knows the alignment. She knows what she did when she jumped on the graph. We don't. We need statistics to infer which path she took. But, she knew where she jumped, and then while she jumped here, she made an A in red and a G in blue. So in the fifth step, she makes an A and a G. Is this clear? Yes. So there is a model, statistical model, and a choice of parameters. And in this alignment problem, we're trying to find the most likely alignment, and that corresponds to the best path from start to finish. So the problem is to determine the most likely path she took, the hidden data, by examining a pair of sequences she generated. So the alignment problem takes this input and finds this output. And that's kind of, again, analogous to the best annotation. Here we're trying to find the best green path. Now, how many paths are there? How many alignments are there? There are 681 alignments in this case. So think about 681 as a very, very, very big number. You cannot possibly try all 681 alignments and find the best one. So it's not possible to examine every single alignment because the number of alignments is very large. There's an exponential number of alignments. 681 is an exponentially large number. And here's a little formula, So if you've taken a discrete math class, so there's a little generating function formula that you can type into Mathematica to find these big numbers, 681. So this book is really a math book, right? It has propositions and things like this. Now, computer scientists know very, very well how to find the best green path, and that's known as a technique called dynamic programming. Biologists, in this context, call this the Needleman-Wunsch algorithm, and the Needleman-Wunsch algorithm, or dynamic programming, very rapidly, in linear time, finds the green path from start to finish. And one way to think about this is there is a statistical model embodied by Diana, and to calculate, to find the shortest path, what we do is we calculate probabilities, but using a generalized arithmetic. Remember, at the very beginning of this talk, we talked about algebra as being the study of generalized arithmetic, but what gets used here is the so-called tropical arithmetic. And this is sort of a sentence, right? The Needleman-Wunsch algorithm is the tropicalization of the pair-hidden Markov model for sequence alignment. Well, maybe just a word. What's tropical arithmetic? Well, algebra is generalized arithmetic. So in tropical arithmetic, the sum of two numbers is the minimum. And the product of two numbers is the classical sum, right? So 3 plus 7 is 3, and 3 times 7 is 10. Okay? So 4 plus 8 is 4, and 4 times 8 is 12. You have to have an open mind if you do math, right? Actually, my kids in elementary school children, actually they prefer tropical arithmetic to classical arithmetic because the multiplication table is a lot easier to memorize. Actually, algebraic geometers also prefer tropical arithmetic nowadays to classical arithmetic because it's actually easier to sort of draw the shapes defined by polynomials. They become piecewise linear, and this is a little bit of this is a tiny picture of a quadratic curve in the plane. Looks funny, but that's exactly what happens. Solutions to algebraic equations become piecewise linear pictures, and arithmetic becomes this funny arithmetic. But that is exactly the kind of arithmetic that we can use to speak about dynamic programming and other kinds of things that gets involved in solving computational biology. Now, why is this a good point of view? Maybe one sentence about this. So this is no big deal in the Google setting. In the Google setting, we have very precise information about the parameters. And in that case, we algebraists have nothing else to say. However, in the biology setting, the parameters are not very well known. And you need to kind of make inference. You need to maybe make statements about the best possible alignment when you don't know very well what the parameters are, and it turns out that this point of view of using this kind of piecewise linear algebraic geometry allows to give new classes of algorithms that in some sense go over a wide range of parameters. Okay, let me slowly wrap up. So the context, the biological context of what I was describing is genomics, comparative genomics in particular. Well, nowadays you don't align just 2 genomes, you align many, many genomes. So maybe there are many sequences, human, chimp, mouse, rat, and you're interested in the so-called multiple alignment. This is an example of a multiple alignment. Again, the stars indicate ultra-conserved bases that are conserved across all the sequences. And this little diagram kind of indicates sort of vertical biology versus horizontal biology. So what I described so far in the sort of gene finding example concerns a single sequence. We might be interested in finding introns, exons, regulatory elements indicated by these colorful boxes. But then at the same time, these sequences share an evolutionary history. So these sequences are derived from an ancestral sequence, and the relationship among these sequences is depicted as a tree. You can see here that there was an ancestral sequence, and these 5 sequences are derived. And you can see evolutionary biology deals with the vertical situation, how these sequences are related, and then the study of a single sequence is here, and the interplay of these two is the field of comparative genomics. So you want to study these genome sequences by comparing them to each other, and if you maybe have some information where the boxes are, then you can use that to find the tree, and vice versa. So the more horizontal information you have, you get vertical information, and things get better and better. Here's another tropical algebraic variety called the Peterson graph. And again, this is a model that describes the possible range of trees that we can have. So I described algebra, discrete mathematics, and statistics as mathematical tools that can be useful for biological sequence analysis, genomics. Now, genomics deals with these kind of sequences, but it's very, very important when thinking about the relationship between biology and mathematics, that biology takes place at many, many, many different scales. So everything I said in the last 45 minutes or 50 minutes took place at the smallest scale, at the sequence scale. But there's organelles, cells, organs, tissues. There's entire organisms like her, but it gets even bigger, right? They're populations. So there are many, many, many different levels of scale in biological systems. And what I described was just a tiny, tiny, tiny piece of the puzzle involving the smallest scale. Right now, at the IMA, there's a wonderful research program going on that deals with biological problems at a slightly larger scale. Now, what's interesting is that different kinds of mathematics have been used to model and study biological problems at different scales. So, for instance, at larger scales, continuous mathematics, differential equations, PDE, dynamical systems have been used. And it's a big challenge for us to communicate. It's a very big challenge for mathematicians to communicate with biologists, but it's also a very big challenge for mathematicians to communicate with each other, who use different kinds of biology to talk about different systems at a different scale. In any case, so this kind of math that I described is relevant at the smallest scale. It will hopefully inform biology, and hopefully we'll be able to communicate with others use differently. Now, every math talk should maybe have a theorem. Every biology talk should have a picture of the lab. And, well, we don't really have a lab in the math department, but these were the students and postdocs that helped write the book. So this is, if you were in Berkeley, up at there's a science museum up there for kids called the Berkeley Lawrence Hall of Science, and there's the DNA play structure, so you can climb on this. And these are the people who wrote the book. Thanks for your attention.
Thank you very much, Bernd and Deanna. We have time for a couple of questions. Any questions for Dr. Sturmfels? Young man in the first row.
Yes. So these repetitive elements, they do have a meaning. I don't know exactly what they mean. The answer is yes. I don't know exactly the answer. That's why one has collaborators. Oh, okay. So the question was, if there are two pieces, how do you know they belong together? That's a very good question. So let me give a very simple answer. So there's something called the genetic code. So the genetic code says that these guys that are boxed, really you should read them in triplets. So three of these guys go together. They form what's called a codon. And then one such triple gets translated into a different alphabet of 20 letters called amino acids. And so really the way to, so if you read from the beginning to the end, you know, it really continues here. When you read this, so they get put together to guys of length 3. And what makes me think, well, that's, this model sort of predicts, that's basically, you know, how it works. That's how the mechanism works. To make the protein, you really need these things, and it has to have, you know, it comes in triplets. So it's one sort of easy answer, the more complicated answers.
Okay, before you go, I wanted to mention, if you're not on our mailing list and you would like to get our poster for the year on postcards, you can leave your address on that form. I want to tell you what's coming up. We have a new kind of event, never before happened at the IMA Public Lecture Series. Next Thursday, November 1st, we are going to be holding the U.S. premiere screening of a film. And the film relates to a picture I have here, to this picture here. That's a famous print by M.C. Escher, the Dutch artist, called The Print Gallery. And you notice it has a white hole in the middle, which has been a mystery for quite a while. And it's really because M.C. Escher wasn't sure what went in there. He had a way to produce that picture, but it sort of fell apart at that point. Some mathematicians, in particular Hendrik Lenstra, from Holland, spent some time analyzing this. It turns out to be very deep. And he solved the puzzle and finished the painting. And it turns out that the whole world is inside that picture. So it's an interesting solution. And that was a subject that caught the imagination and other aspects of the math and M.C. Escher's art of a Canadian documentary filmmaker named Jean Bergeron, who made a documentary on this subject, a 50-minute documentary for Canadian television. It's going to have its world premiere on October 24th, so you might want to fly to Montreal for that. Or if you can wait a week, it'll have its second showing and its U.S. premiere here in this room as part of our lecture series, and Jean Bergeron will fly in and take questions at that time. So that'll be our next event. I hope to see a lot of you here. We have two great ones coming up after that. In February, Alfio Porteroni is going to talk about math modeling in medicine, sports, and the environment. I won't say much about it, but Alfio was the math modeler whose team helped design the Olympian racing yacht that took the America's Cup trophy away from America and put it in landlocked Switzerland, where it remains. And then we have Ivar Eklund, who is going to talk about the best of all possible worlds and bring the idea of optimization from the Enlightenment and the Renaissance up to the optimization that happens in places like Google and Amazon.com and the U.S. Congress and so forth. So I hope to see you back here for those. Thank you for coming tonight. Good night.